Now, what do we have next in reproduction and development of angiosperms? What lies here drawn is the structure of anther. And what we have to study is anther structure. Anther was part of stamen, that is part of androecium, and we have to see the transverse section of anther. Why do we study transverse section of anther at anatomical level? So that you get an idea when we talk about the microsporogenesis in coming lessons, you have an idea what is taking place where. So in the structure of anther, you know that supposedly this is the entire anther like this, bilobed anther with the filament. If we cut it from here and watch it, observe it supposedly. Now this would be anther like this, something like this, okay? Something like this. Over here you see 4 pollen sacs, okay? This is what, and the remaining of it would look like this, and you have this structure. Now this has been magnified and seen over here. We have layers of cells and we name it one by one. Outermost, as you know, it is epidermis. The one being represented by color green is epidermis, followed by large sized cells. The layer of these cells is known as endothelium. Endothelium is followed by few layers of cells which are known as middle layers. These are the terms that are to be remembered. And what you see over here is that there is a connection between 2 regions. This region is, this part is known as connective. Okay, this is the connective region which joins the 2 lobes of anther at the back, known as connective. This blue part that you see over here is vascular strand. Inside the, I'm going to write it over here like this, vascular strand. Okay. So by now what we have studied is that there is an outermost uniform layer that covers the anther known as epidermis, followed by a layer of cells known as endothelium, which is followed by the middle layers, and the red part that you see over here that surrounds the green part is known as tapetum. This layer known as tapetum has a nutritive purpose. Inside tapetum we have the sporogenous tissue. Okay. Now you see these 4 circles. These 4 circles are nothing, this entire red part. These are sporangia. Now what are sporangia, otherwise known as pollen sacs? These sporangia are the site where sporogenous tissue lies. In each sporangia you are going to find this sporogenous tissue. This sporogenous tissue would undergo meiotic division and form the pollen grains. That is why this each region is known as pollen sac. Okay? This region is known as pollen sac because ultimately it is going to form the pollen grains. Otherwise the name for this is sporangia. So what you see over here is that anther is tetrasporangiate. You have to remember this. Tetrasporangiate. Sorry. 4 for tetra, sporangia, and here I am writing it wrong, tetrasporangiate anther is there. Bilobed tetrasporangiate anther which has a vascular strand in connective that connects the tetrasporangiate anther bilobes at the back. Okay. So this is the connective vascular strand, tetrasporangiate anther. We see its transverse section. Inside each sporangia lies the sporogenous tissue. This sporogenous tissue would later on undergo meiosis and form the pollen grains. Now this is a fresh anther. Supposedly this anther has matured, what happens over here is all these middle layers are gone, endothelium is left little bit, the vascular bundle is almost consumed, these things are totally consumed, and these sporangiate walls are gone. What do you find over here is 4 pollen sacs and the pollens which are spread uniformly inside it. So you are going to find pollens over here, pollen grains, alright. Those pollen grains are formed by a long process known as microsporogenesis, and you find pollen grains over here. Now what happens over here is that this epidermis is stretched and broken down after some time and it forms stomium. Okay, this empty space is known as stomium. This stomium later on bursts and pollen grains are thrown into the air. So this is what you have to remember in the structure of anther. We had seen that the transverse section of fresh anther had these things which I have rubbed now, but you have to remember outermost was epidermis followed by a thick layer of endothelium which had subsequent layers of middle layers, and these layers stopped at tapetum, which was a nutritive layer for the sporogenous tissue which was present inside 4 different sporangia inside the 2 lobes of anther. Later on when the anther develops, when the pollen grains are formed, this entire middle layer and rest of the machinery is being consumed. Now I would not say machinery, rest of the arrangement is consumed and pollen grains occupy the entire space. Now these 2 points, this outer layer forms the stomium and it opens up, okay, it dehisces. Also this acts as line of dehiscence, this could be line of dehiscence and it is going to burst and the pollens would be given out in the air and then it could be taken from one place to another, that is known as pollination. So this is you have to remember in the structure of anther. When you click on to the next lesson, you're going to see that what we had represented as these simple representation of pollen grains, how these pollen grains are formed at the level where we see the pollen being formed from the individual tissue known as sporogenous tissue getting converted into the microspores.